Yes, yes, thanks a lot, first of all, for having me here and giving me this opportunity to present this work. So my name is Wali Inam, and I'm the PhD student at Akademi, working in Hong Kong Zhang's research group in PSL laboratories. So this is today I'll be presenting this work, which is a part of my PhD, and basically it's about how polymer particles can interact, or buffers can interact with the polymer particles, and how it impacts the zero potential measurements. And yeah, so I will first start with some uh, basic stuff, just to build my point. So basically, nanoparticles, since we were mostly talking about the interaction of the particles with the buffer. So first, we'll look at the particles. So simple definition. So these are the materials which are containing the particles in the range of 100 to 1 to 100 nanometer, which may be in unbound state or agglomerated state, uh, uh, will be called as nanoparticles. So basically, nanoparticles have two regions, or two. We can divide them into two parts. Even a simple polymeric particle, if we talk about. One is the matrix or core, which we can call as, and other is the surface. So the matrix and core, matrix and the surface may look chemically same, but they at least differ in one aspect, that in the matrix is actually is not directly exposed to the exogenous molecules. However, on the other hand, surface of a nanoparticle is, it can directly interact with the molecules which are in the solution, or exogenous molecules. So so therefore, since it can freely interact, so what we talk about is the particle surface when interacts with these molecules. So it interacts with the help of certain functional groups which are expressed on its surface. And these functional groups can attribute certain characteristics to the surface of the particle. So for, let's say, they can attribute certain charges and can express certain charges on the surface. So depending upon the type of the molecule, or whether it's a carboxylic group or amino group, let's say, a particle may possess a negative charge or a positive charge on its surface. Now, when these particles are in the solutions, so since solution have ions, in a particular active solution that I'm talking about, then because of the charge which is possessed by the surface, some layer of ions are directed from the solution are directed towards the particle surface, and this first layer of ion is known as tunnel layer. Now, since in the environment, or in the nature, things tend to move towards the equilibrium. So naturally, the other ions in the solution, they gather around the particle, and they form this kind of a cloud of ions around this particle, which is known as electric double layer. Now, this electric double layer extends to the point where these ions further no more adhere to the particle, and they are kind of this is the last boundary where the particles adhere to the nanoparticle or particles, and this particular point is known as slipping plane. And this is at the point where we measure this electric mobility, or electric potential of a particle, or which is also known as termed as zero potential. So when these particles are placed under some electric field, so because of the potential possessed by these particles, they either move towards anode or cathode, and this mobility, electric mobility, is measured, and which is then used to calculate the zero potential. And this electric double layer and zero potential is influenced by many factors, particularly two: pH and ions, concentration of ions of an aqueous solution. So, in order to measure this zero potential, we have to keep these two things in check: pH and ions concentration. For that, commonly buffers are used. But buffers itself are they are more diverse in their structures. So they may also possess certain functional groups expressed on their surface. some cationic centers and anionic centers in order, because their purpose is to to resist the change in pH. And on the other hand, we have also functional groups which are expressed on are present on the nanoparticle surface. So what the question is, if the possibility, if these buffers can interact with nanoparticle surface, and in doing so, if they can affect the electrokinetic properties of the particles. So we were interested to see that how this can occur, what type of interactions can exist, and if they exist, for example, then how it can affect the electrokinetic properties of nanoparticles. For that, what approach we adopted, basically, I made these polymeric nanoparticles and use certain set of buffers, and I studied these at least two things. For example, the Zeta-potential was studied for them, and also we did some computational modeling. So we use Zeta-potential as a basically the main component or main parameter to see as a main parameter to measure the electric kinetic property of the particles. So zero potential, since zero potential was the main parameter which we were trying to measure, and as also as a kind of indirect measure to see how particles will interact differently with different buffers. So you studied zero potential three parameters, and the three parameters: pH, buffer concentration, and reaction ratio. Now, pH is important when we're talking about as a journal as well, because pH tends to influence the state of ionization of a molecule or a functional group. So on one end, if we go to the acidic part of it, for example, if we go to the acidic pH, then we can see that amine-based groups can get protonated, and then on the other hand, they could be a deprotonation of the carboxylic groups, and in the middle we have this ionic form where both kind of ions are contributing to the same molecule. So what if we change the pH of the solution, and then it should cause same a change in ionization states of the functional groups which are present on the particles and the buffers. And then if the change in ionization is happening due to protonation, deprotonation, then molecular interactions, how these particles and buffers may interact, if they're interacting, might change. And that the idea is if that can be reflected in the zero potential. So to do that, we took two buffers, types of buffers. One HAPPIS, commonly used buffers, HAPPIS, which is contains two cationic centers and one anionic center, and one tris with only one anionic center. And then we used 6 polymer types or polymers, and check the zero potential of these polymers under range of pH. So what we did, we made a comparative analysis of the HAPPIS and used to measure zero potential of each of the polymer particles, and we use a range of pH to do that. So here I will particularly focus on to make the short my talk a little bit concise. I will mainly talking about ACDEX, SPCDEX, and PLGA. Now, when as the first we did the comparison between the HAPPIS and tris using ACDEX. So ACDEX is basically the dextrin-based polymer, and it has these acetyl-based functional groups, which makes it pH responsive. So these acetyl groups are hydrolyzed at acidic pH. So when we were trying to measure the zero potential, so we were not very concerned about this part, because here the hydrolysis of nanoparticles might start. So what is was important to see in this kind of a comparison, that first you can see that there was a suppression particularly in the trace space, in the trace zero potential of the particles in the presence of trace. And particularly there's the inversion, like where you get a slightly positive zeta potential. However, in the Hapis, there's a strong or relatively strong zeta potential with Hapis. Now it has SEDEX has a hydroxyl group, and it is expected to present or to have a negative zeta potential. So this was something which we were not expecting. And then secondly, similarly in the PLGA, what was observed, the quite strong suppression of zeta potential, so it remained relatively constant. However, in the presence of trace, while in Hapis, there was the zeta potential was quite so it was relatively higher. And also, if you take the same pH range, so you can see a significant difference was at 5.5 or 6 pH. The reason, and so we what we thought that particularly probably the trace is probably doing some interaction with the nanoparticles, and that's causing this suppression or inversion of zeta potential. So when we did the empty simulation studies, and we tried to figure it out, particularly we were looking for a hydrogen bonding interaction between these buffers and the polymers. So we clearly found out that trace has the probability to make more hydrogen bonds with this ACTX polymer, and also with the PLGA, the number of hydrogen bonds even they were more with the trace. So this we speculated that these, because the trace has the more probability to interact with the Hapis buffer, so it kind of suppresses or caps the negative zero potential, or caps the negative charge groups which are expressed on the ACDX or PLGA, particularly in PLGA where it has a cup stick group at the terminal end. So this was the case. Similarly, we also worked with the another kind of polymer, which is SPACDEX, which is the derivative of ACDEX, because it has this chain of spermine chain, which has a lot of anisotropy groups expressed over there. So therefore, this gave us the opportunity to see the alternative, that what happens if we have amino groups there. So what we saw that again in trace there was a little bit suppressed zeta potential, but particularly what was interesting to see that at a little bit alkaline pH, and there was kind of inversion of zeta potential. So slightly negative zeta potential was observed. However, if you see the structure, we expect zeta potential to be a little bit positive because of these spermine chain, but this inversion was observed. And this can be correlated also with the number of with the empty simulation studies where we found the Hapis has the probability to make more hydrogen bonds. And if you see the structure of the Hapis and observe this, it is a little bit alkaline pH. So there is a chance that Hapis will get deprotonated from the sulfonate. Sulfonate moiety will get deprotonate, and this will increase the chance of interaction of Hapis with the buffer, and thus it caps the positive positive charges and somehow expresses slightly negative zeta potential. Now, the second thing which we were interested in to see the concentration effect of concentration. What we know from the literature as well, that okay, in low ionic concentration, the Debye length is higher, and which leads to higher zeta potential. However, in higher concentration, this is Debye length or the electric double layer decreases, and also there will be therefore a lower lower zeta potential. This is not high, sorry for this, but lower zeta potential. So what we wanted to see, that if we change the concentration of the buffers, we analyze the zeta potential of particles over a range of buffer concentration, can we see the change in zeta potential, and that may reflect the interaction between the buffers and the nanoparticles. So what again we did, is we use hapless in trace buffer and measure zero potential over range of buffer concentration from 5 millimolar to 20 millimolar. Now, and this also we saw some very interesting things. For example, in the case of SPCX, first, if you see that zero potential remain of the SPCX remains relatively constant over hapless and trace. But then there is a decline in the zero potential, which is expected because we are increasing in concentration. But what is surprising or very interesting to see that in the trace, there is a more steep decline in zero potential, while on the other hand, in the hapless, the zero potential was not suppressed that steeply, and it was kind of didn't change that much. However, again, if you follow ahead, that they're at 50 millimolar, they again start to match it. Their zero potentials start to match with each other. So something is happening at this point, which is making something zero potential of SPX in hapless different from the trace. Now, what we found out also is from the MD simulation studies as well, so what it feels like here that hapless SPS in the presence of hapless, the zero potential is more getting stabilized, which means it has the one anionic center, the sulfonate moiety, and it can, as simulation studies also show, and we have seen from the hydrogen bonding studies as well, that it can interact with the positively charged group or these fermion chains on the sp6. So this interaction possibly promotes the absorption of these hapless molecules on its surface. So since it has two cationic groups, so it is kind of adding to the surface charge of the surface of the sp6. Thus somehow, so therefore neglecting, somehow resisting the change in the potential by slightly strengthening the surface charge. Hence, because since we are increasing concentration, so absorption will still be there, but at 50 millimolar, there is probably a lesser absorption or no more further absorption, or not enough so that it can overcome the higher concentration which is outside of the ions, and here and therefore we can get at 50 millimolar, the both trace and they start to behave similarly. And this with MD simulation studies was possible to explain when you think of the number of hydrogen bondings which can support this interaction, and also charge-based interaction. Similarly for the ACDEX and PLGA, we made the comparison, and what we found that there was kind of an inversion of zero potential which was observed at higher buffer concentration, and this also explains that because it has a one cationic center which can interact with the carboxyl groups on the PLGA, and thus it can also inversion means there's slight absorption of trace, and it is kind of capping the negative potential of PLGA. And also, I explained earlier that trace has a possibility to form more interactions with the PLG and ACDEX as it happens, and that explains the reason for the results. Another thing which we did was we did the experiment where we made the fraction ratio between the nanoparticle and buffers, in which we mixed different concentration of nanoparticle buffers in a way that we made higher fraction ratios between the nanoparticle and buffers, so that there were high number of nanoparticles but low number of buffer molecules. But gradually we decreased the nanoparticle concentration and increase the buffer concentration, so that we can observe if there what changes are brought at the surface of nanoparticles that were read by the zero potential measurements. So first I will try to explain the ACDEX. And ACDEX, what we are basically doing here is we are kind of decreasing, when we are going here, we are decreasing the nanoparticle concentration and increasing the buffer concentration. So this is a lower buffer, little bit lower buffer concentration. So here you can find a little bit negative zeta potential, obviously because of the hydroxyl groups with the acidex. And as we go down, there's a decrease in zeta potential that was a little bit obvious, and overall shift was 9.8 millivolt. There was nothing surprising in that. We expected, okay, we are slightly changing the buffer concentration, so probably it is as a result of that. But what was interesting to see in the case of SPCDEX, and that when similar experiment was performed, there was a huge shift in zeta. So what happened that we get very high zeta potential, obviously because of these amine groups. But as we start to decrease the nanoparticles and increase the buffer concentration, we get a very huge change in a shift, 43.5 millivolt, and at lowest fraction ratio, we got minus 14 millivolt zero potential. That was a little bit surprising because we expect to have it still positive because we have these permine chains on top. So here was definitely the question what was happening, and definitely that what we found, obviously to have simulation studies as well, that okay, here as well there's interaction between these amine chains and the permine chain and this sulfonate moiety because of the ionization thing, and then therefore it caps this its zero branch, its charge, and we have on the opposite hand hydroxyl group, which may lead to this negative, slightly negative zero potential which is there. We in them similarly in the studies we were able to see that if you see these, the green are the charge groups which are present on the polymer, and this blue or cyan color it represents the buffer. This one, and there's in the yellow you can see the interactions which are happening, hydrogen bond interactions via the water molecule. Again, we did the same experiment for the PLGA, and in the PLGA, we again observed a huge shift. Now, this shift actually couldn't happen only because of the buffer concentration, because we very slightly changed in this fraction ratio. The buffer concentration was slightly changed, not that much, only probably 20 millimolar change. At a higher fraction ratio, because of the hydroxyl groups have closer to minus 60 millivolt, and when we go to the lower one, then we get this zero separation, which is around 47.3 millivolt. And this can not only be explained by the change in by the slight buffer concentration change, but explains that somehow these piperazine rings, which are associated with the Habeas molecule, they are interacting with and capping the negative charge of the PHA polymer or these particles. This is the overall for fraction ratio experiments, that this is how we observe the shifts in different kind of the polymers, as we can see. So also in other type of the polymers, which are HPNCS polymers, the three years we use. So similar shifts were observed. And then I will just give you an overview of what we were trying to do and what we conclude from this. So buffers can definitely interact with the nanoparticles, and this interaction can be supported by electrostatic interactions, hydrogen bonding, and van der Waals forces. We mostly focused on hydrogen bonding, but in the simulation we also found van der Waals forces, other type of interactions playing their role, though we didn't discuss here. And this interaction can lead to at least three type of the patterns we can observe. One is the suppression. We can observe zero suppression because of the charge capping. Number two, what we can observe is the stabilization, that at higher as we are increasing the concentration of the buffers, it can lead to absorption of some cations and can stabilize the differential or prevent its decrease. And then inversion, which means that somehow when particles then buffers they interact in a way that they also they orient in a way that they express their negatively charged groups or hydroxyl or carboxyl groups on the contrary on the top, so that because of the particular orientation of the buffers, we they also cap, but also the alter or invert the zeta potential of the particles. So I will just conclude now by saying that the take home message or the conclusion which I want to emphasize here, that that the buffer selection of the buffer is very important thing for the operational measurement. So that should be carefully done. And then what in this paper we have done is provided alternative approach to measure the potential, that is through fraction ratio based measurements, which are better and way to measure the potential as compared to single point zero potential measurements. Further, this idea can be used to study more broad range of the buffer molecules, to see what other structures can influence, what other structures can structures are important in the buffers which can influence the electrokinetic properties of nanoparticles. And such kind of studies can be studies can be made with a small drug molecules, and data can be used to predict the drug loading and drug release profile. With that, if anybody wants to read and get more insight about the article, so this we published recently in the Pew Journal. And and definitely thanks a lot for listening, and I'm grateful for my supervisors, Jessica and Hongkong, for giving me the platform or guiding me through. And a lot of thanks to Sergey, because he really helped me to understand the electromobility, the differential data and everything. And also the Rajendra for doing the MD simulation studies, and a couple of master students who contributed in here as well. And yeah, I'm happy to now take questions.